All right, looks like we are live, and I want to welcome everybody to Standing for Truth. My name is Donny, and I am your host for tonight's program with Professor David McQueen on Paleobotany. Did herbivore dinosaurs attack rose bushes? David has been doing some fantastic research on this topic, and I am excited to see what he is going to present tonight. David, my brother, always a pleasure, always a privilege. How you doing tonight?
Doing well. We often start off with some of George's humor, but I think we'll get more serious with a Shakespearean quote. What is that in Shakespeare? A rose by any other name? And so we're going to see if the evolutionary community, with their actually comical idea that thorns on rose bushes like this from Ms. McQueen's garden, you see the thorns there, that they actually suggested to me that they evolved to keep herbivores from eating the roses. So I went out to get some roses from Mrs. McQueen's garden, and here's some nice edible roses for the herbivore. And you actually have to move down the stalk pretty much to see the thorns. For example, I've got my finger there on a thorn. It's kind of hard to see. But the very idea that the rosebush could know through natural selection that it needed to fend off the herbivores started me on this path to study paleobotany since that ridiculous suggestion was made. So I'm glad to be here, Donny.
Well, I am glad to have you, David. I'm glad you've come handy with your gloves. We don't want any injuries during today's show on paleobotany. So,
how true.
Great introduction to everybody in the audience. Yes, as usual with these programs, we will be interacting and engaging with your questions and comments. And so if you've got questions, objections, comments, please tag me. I will save them and we'll have David address them. Real quick before I hand it over to you, David, I also want to remind everybody, if you want to see more from Professor David McQueen, please do check the description box. I've got two separate playlists. One playlist with all of David's past presentations with us, and another one with all of your debates, Professor McQueen. So you've done a lot of debates, and I appreciate that.
I am so thankful for Standing for Truth and for you, Donny, because from looking at my own notes, we're coming up on a 2-year anniversary from the first time that Samuel Church saw a presentation I did and recommended me to you. So I'm thankful to be coming up on that 2-year anniversary.
Well, you've been a true blessing, and you've done a fantastic job defending the truth of biblical creation. And David, that's what we do here on Standing for Truth. So let me hand it over to you, brother, for your talk on paleobotany.
Okay. And Donny, if you'll help me and set a timer for about 20 minutes,
sure.
I'd like to go for the first set of questions in about 20 minutes. Okay.
Sounds great.
Let's get started as we always do. Remember that we're defending biblical creation. And so I would encourage you to get your copy of the Bible and look back at Genesis chapter 1. And you need to think about it in the context of the Garden of Eden. And so when we think about the creation of plants during the creation week, we have to also be mindful that plants, flowers, fruits, nuts, contain all the basic amino acids that we need as humans. This is why, even in a fallen world, a vegetarian diet has been followed by many people. You have heard me on other occasions talk about my work with Robert Gentry on the radioactive halos. One thing that you might not have heard me say is that Bob Gentry was a very committed Seventh-day Adventist, and one characteristic of a committed Seventh-day Adventist is the focus on a vegetarian diet. Now, Bob Gentry, when he would invite me and Shirley to his house to eat and so forth, they would create like a lasagna, for example, that tasted like it had meat in it, but it really didn't. It was some kind of soy or nut. And Bob Gentry pointed out to me that, this was the basic plan of God. Initially, Adam and Eve were vegetarians, and it wasn't until later that we read about people eating meat. Leviticus 17:11 comes to mind. For the life of the flesh is in the blood, but it's the blood by reason of the life that makes atonement. And so we do deal with blood sacrifices and the eating of meat, but that was not the original plan. Bob Gentry pointed out to me in my friendship days with him when he and I would talk about things other than radioactive halos. He pointed out to me that in a fallen world, in order to get the correct number of amino acids and so forth, you have to be very careful about your diet. For example, you can't just eat a cabbage salad. You've got to supplement it with certain nuts like cashews and almonds that have in them the additional amino acids that our body needs. And so he was a very careful vegetarian. And one of the things that he loved to point out to me when we would visit his house in those days is he had a German Shepherd that he also fed a vegetarian diet to. And this German Shepherd had a shiny coat and real nice looking dog. And so it's clear from what the Bible teaches that God created those plants during creation week. And if you read the passage very carefully, and I may, after our 1 hour break, come back and bring a copy of Scripture to read it to you. But I think it's something that you will have read in the past, that God created the ocean, and then the rocks rose up out of the ocean. And the first thing that he created was plants. And he goes into quite a detailed description there in Genesis chapter 1 about how the plants fall into categories. There are herbs, there are fruit trees yielding fruit in their season, and so forth. So that as we move up to the Garden of Eden itself, we see that Adam and Eve were able to function quite well, and the animals too, for that matter, on a vegetarian diet. One of my favorite stories about this very thing comes from my interest in World War II history. And when the German army fell short of meat, and they regulated their diet by how much fat they would get, German sausage kind of stuff. During that time, they fed the animals in the Berlin Zoo a vegetarian diet. And so those lions and tigers and so forth in the zoo during World War II did quite well on eating that vegetarian diet. In previous debates with evolutionists, they've turned to me and said, "Oh, McQueen, how do you explain like a a vampire bat, or how do you explain the saber-toothed tiger teeth and so forth? The creation scientist, Dr. Gary Parker, who's now retired in Florida, he had a saber-toothed tiger tooth that he would use in his lectures. And he pointed out that the creature probably used that tooth not to eat other animals in the garden, but rather to bite into mangoes and fruit and so forth, and use that tooth to be able to get that kind of fruit to eat. Furthermore, he said in his study of the anatomy of like a saber-toothed tiger, for example, if you were to pull hard on one of those teeth, it would come right out, because it was never intended by God to eat meat, but rather to forage and get fruits and vegetables. Now, the argument that we want to address tonight is an argument from Paleobotany. This is an area of study that people like John Mackay in Australia has spent a lot of time studying. The members of Standing for Truth that are very interested in nutrition certainly have seen the value of plants. But here at Standing for Truth, we love to see weak arguments presented by the evolutionary community. And so, just to reiterate a comment that was made in one of my previous debates, let me put my rose garden glove on here and pick up this rose here. Get myself organized so you can see the argument. So it's got a vine growing around it, but you can see that at the top of the thing, there are some of the roses from Ms. McQueen's garden. Remember, I'm broadcasting from Louisiana, where the ambient temperature outside right now is 100 degrees. So, you know, if you like flowers and so forth, this is the place to move to. And you'll notice that there's a vine associated with it. But the very idea that when the rosebush recognized that herbivores were eating the roses from the top, natural selection evolved thorns to keep those herbivore dinosaurs from attacking the rose. Now think about how foolish that argument is. So what the evolutionary community is saying is that these kinds of thorns, which on a rose bush technically are not called a thorn, as I've studied the botany of this, they are a different, they originate from a different area than the thorns on other bushes and trees originate. It's kind of coming from the epidermis. But the very idea that the rosebush somehow knew that the herbivores were going to eat the roses, and so they evolved these thorns to keep the herbivores away. Well, let's notice the foolishness of that argument. Notice in what I picked just a few minutes ago, the roses are at the very top. An herbivore dinosaur could have snacked on this and never gotten down low enough to even have to worry about the thorns. But the very idea that this God of natural selection would allow a rosebush to understand that it had to evolve thorns to keep this dinosaur away is ridiculous. It's logically ridiculous. And if you look in the literature, it's even more ridiculous, bordering almost on comical. Let's make sure that we see the evolutionary argument just from what I was able to pick from our backyard a few minutes ago. This is from our tulip poplar tree. And let me take one of these off so you can see where the word comes from. I hope you can see that the leaf itself looks kind of like a tulip, but it goes beyond that. As the life cycle of this tulip poplar goes through the year, there are real pretty tulip-shaped flowers that fall off this tree, and we can pick it up. So the evolutionary argument is that a deciduous tree like this evolved over time from something like a fern. So here we have the fern from the garden, our garden, and then here we have the deciduous trees. When I have prepared for this talk tonight, I have actually tried to find discussions in the literature of the evolution of plants. And in my research, I went back to documents and references from the 1940s and 50s. And the evolution of, like if you look up flower, for example, it doesn't talk about the evolution of flowering plants. Not until you get up until the 21st century do you begin to see depictions of the evolution of plants. And often what they will do in these references, they will show a modern forest, a conifer forest, evergreen forest, and a deciduous forest. And then in an earlier panel, they will show from the fossil record, predominantly in coal seams, they will show lycopods and other plants that they feel the gymnosperms and the angiosperms evolved from. At the University of Tennessee 50 years ago now, my paleontology professor kept a plant in his paleontology lab, which was supposedly what we in Standing for Truth would call a living fossil. It was a type of plant kind of related to a fern, if you will. But he had this thing growing in a pot, and he said, this is didn't use the word living fossil, but he talked about it being a fossil nonetheless, and tried to portray what it meant. In my study, I've gone back to this book, which I would recommend to you guys. It's a book called The Fabric of Geology, and it was put out by the Geological Society of America back in 1963. Now, you have to understand that during this time, there was a tremendous movement among conservative Christians. Now, this came out in the 60s. Dr. Henry Morris began his lecturing in the 1950s. He and Whitcomb published the Genesis flood that's funny about Smash Those Roses. The evolutionary community wanted to have a reaction to this growing argument that evolution was simply not true. And so they found famous scientists like George Gaylord Simpson. He was one of the leading paleontologists of his day, and he wrote a very long essay in this book that I am referencing here that deals with evolution and how it's a true historical science and so forth. But it's very interesting to see what he says in the book when he begins talking about the evolution of plants and so forth. And he says this. Points always at issue in historical science, this is from page 43, are the consistency of proposed laws and properties with known historical events and asking, is it sufficient to cause evolution? Probably the strongest argument that the catastrophists had, now he's talking about the viewpoint I've come to as a flood geologist, but also the viewpoint that was prominent in science all the way up until around 1700. He said, probably the strongest argument that the catastrophists had was that the known features of the earth were inconsistent with their formation by known natural forces within the earth's span of existence, which many of them took to be 6,000 years. That is my argument too. The fault, of course, was not with their logic, but with one of their premises. The same argument with the same fallacy was brought up against Darwin when it was claimed that his theory was inadequate to account for the origin of present organic diversity, then estimated by most of the people in 1859. They thought the earth was maybe a few million years old. Darwin stuck to his guns and insisted correctly that the calculations of the age of the earth must be wrong. Can you see that the argument that George Gaylord Simpson gave about Darwin is very much what we've talked about in the past? Now, in this essay in 1963, he talks about something that he calls Dollo's Law. And that is worth writing down up here because it was new to me in my study of these things. I'm curious to know how many of you guys have even heard of this thing called DOLLO, Dollo's Law. And this scientist proposed this. He said, evolution is irreversible. And so we never can go back. We always go forward. Now, the work of Donny and others that have presented on Standing for Truth, you can clearly see that the problems in, for example, endogenous retroviruses, they are real problems that don't go away when you try to go back and present it another way. Well, he introduces a law that applies to plants also, and it's called Cope's Law, C-O-P-E, Cope's Law. And the idea of Cope's Law is every organism evolves with time. Every organism, by the O there, evolves with time from small to large. And so,
David,
yes.
have you heard of coleslaw before?
Oh, coleslaw, like you eat at supper?
Right, yeah.
Yeah, very, very.
Not a big fan. Okay,
That's
go ahead.
Canadian humor there. Thank you. Am I close to my first time break, by the way?
Oh, you're doing great, David. Let me just double check real quick. Saving questions. Yeah, you've got about another minute left, but no,
Oh,
no
okay,
pressure.
good.
Take your time.
I'll end it this way. If you go to a scientist like George Gaylord Simpson and you actually read this Harvard biologist biography, it turns out that George Gaylord Simpson grew up in a Christian home. And it wasn't until his teenage years that he became an agnostic. From my conversations with evolutionists, whether they're talking about the evolution of from trilobites to dinosaurs, or when they occasionally talk about the paleobotany, their standard argument is, oh, somebody like George Gaylord Simpson was brainwashed by reading the Bible. Our argument is that he was illuminated in those years, that he was a Bible student and then changed. Donnie, back to you now.
David, excellent stuff. I appreciate the presentation. I looked over at the timer and I couldn't believe that we were already at the 20-minute mark. So time flew by there. We've got a lot of questions. We got a fun chat tonight.
Good.
I wanted to put this one up. Taylor K is laughing at the Canadian humor. In other words, not very good humor.
Well,
But
you guys, half your country apparently is on fire and you got smoke blowing. And so you have some problems, don't you?
It's rough. It's rough. But Redefine Living does say Donnie's law. So my law is to the evolutionists, don't bring your assertions or opinions, bring your arguments. And we want those arguments to be backed by evidence, right, Professor McQueen?
Well, very good. Very good. So let me have one of those questions from the audience. I'm interested.
Okay, here we go. We'll start from based on the points you started with in your presentation.
Yes.
And so here's the first question. Critics, I've specifically heard this one many times, so I think this is a good one to address. Critics on Talk Origins, for a little context, talkorigins.com is a prominent site that is very much anti-young Earth creation. So the critics go there constantly. Okay, so they say critics on Talk Origins have argued that Dr. Gentry is unable to demonstrate that the concentric halos in mica are caused uniquely by alpha particles resulting from the decay of polonium isotopes. What's a good response to that criticism, David?
Let me move my board down here and go over to the whiteboard and refresh everybody's memory about some important details about polonium halos. First of all, let's make sure we're clear on the chemistry of what we're talking about. Uranium decays to lead, and as it decays, it goes through a variety of elements, one of which is the element polonium. So this is not Pu, no, it's not plutonium, but it's polonium. And one of the isotopes that is of very much interest in polonium halos is called polonium-210 and polonium-214. Okay. 50 years ago, when I worked for Bob Gentry at Oak Ridge National Labs, the job I had was to take a piece of fluoride, calcium fluoride, that mineral, or mica, biotite mica, and I would take a Q-tip and move it over the surface of this thin section, and I would sand down until I got to a point where the halo was truly the diameter. Now, why would that be important? If you imagine a sphere and you begin to make concentric cuts through the sphere, you're aware that you only get a true diameter of that sphere if you are right at the center of it. And so this distance here, the diameter of the halo right at that point, that diameter through probably well over 100 years of study, what Gentry calls a radioactive halo, geologists call a pleochroic halo. It has to do with a color change under a petrographic microscope, cross polars and so forth. So at any rate, this pleochroic halo, or what he called radio halo, this diameter here in the geologic literature was assumed to be constant. And since the decay of polonium follows the decay laws of radioactivity, the decay rate, which is called lambda, the decay rate lambda was always assumed to be a constant, so that the radioactive decay rate of U-238, 4.5 billion years, and so forth. Well, what Gentry realized is that some of these isotopes of polonium have a half-life of less than 1 minute. Now, if you find these in granites, where the mineral biotite is in the granite, and then you do an analysis of the halos in the biotite, the mineral is biotite like this. If you do an analysis of that, what Gentry found is there is variation in the diameter of that polonium halo. Now, the variation is such that it leads to two conclusions. If you give the evolutionary community every benefit of the doubt, it turns out that lambda still has an error bar in it, but the error bar allows it to be 38% accurate. And so some of my critics over the years, when I presented this, they said, well, McQueen, if you're talking about a 30% difference, it's not going to change a 5 billion year old universe down to 6,000 years. And that's true. But they don't study Gentry's work very carefully. He goes on to say that if you do not give the evolutionary community latitude and you actually work through the differential equation, d lambda over lambda, and you work through the calculus and differential equation of the thing, if you have finished that analysis in a way which would favor a rapid creation of the universe, then it turns out that you can say nothing about the constancy of radioactive decay. And so Gentry's approach to this was always, from the 1970s on, Gentry's approach to this was always that if you have a different cosmology, if you have a different assumption that would allow the rapid formation of the universe, what he called a fiat creation of the cosmos. If you have a different viewpoint, that changes the way that you come to understand radioactive halos. I hope that answered the question.
Yes, it did, and I appreciate how comprehensive you were, David. I want to try and to represent the next argument correctly, but I'm curious if you've heard the critics argue against the polonium halos argument in Dr. Gentry's work by saying his samples are not from primordial pieces of the Earth's original crust, but basically from rocks that have been extensively reworked from other areas. Have you heard that? And,
Oh,
and
yes.
could you speak to that a bit?
And I have discussed this with my creation colleague, Dr. Andrew Snelling, who is worried about where some of these rocks came from. Now, the geologic word for where a rock is found is called the provenance. So if I take you to see some plant fossils in Middle Tennessee that are polystrate trees, the provenance of that is those rocks. They're supposed to be Mississippi and Pennsylvania. They're in central Tennessee. Well, an argument that many have used about Bob Gentry's samples is that he did not make a chart in one of his papers that actually showed where each of the samples he got came from. Well, when I was working around him in the early 70s, I was going through my undergraduate degree in geology, so I recognized the importance of this. But he minimized it. He traveled to Europe to get some samples from a well-known European locality called the Wolfendorf pegmatite. And so he got these samples from Europe, which he actually went and got with his own hands. But he brought these back and did the analysis of the biotites and the fluorites from this European pegmatite. But one thing he never did, which is a legitimate criticism of Gentry's work, is he never listed the 10 most important samples he used to do his research on and tell exactly where they came from. Now, as the years went on in the early 70s, Gentry, I think, began to recognize the importance of this because at a certain point, he got some samples from the western U.S. from what are called roll front uranium deposits. Now, these are deposits in sandstone where uranium rich waters moved through. That's why it's called a roll front. And these waters moved through. And as they moved through, there would be wood samples that during the flood had begun the process of coalification. Well, when Gentry looked at some of these wood samples, he recognized that there were halos in there that were deformed. They were not circular, but they were elliptical. And he recognized that these were secondary halos that where the polonium had moved through, carried in an aqueous solution, moved through this wood, and then deformed by events of the flood, and they were elliptical in nature. And he looked at the lead ratio in this coalified wood and compared it to these samples from pegmatites. And he was able to show that the lead ratio is different here than there. Put another way, he silenced his critics by making it clear that he could tell the difference between what he called primordial lead ratios and something that would be related to Noah's flood. Next question.
And so basically the primordial ones, those would be the result of the initial creation event. the
Exactly.
secondary halos during the flood.
That's exactly right. Yeah.
Okay, very good. I appreciate that, David. Great job. Okay, next question that comes in is from Doki Doki Bible Club. Thank you very much, brother. At David, do thorns protect cactuses from being eaten?
A very good point. As I have studied in the last 30 days, this whole idea of the difference between a thorn, a true thorn on a cactus, for example, and what I have on this rose bush here. Let me get down to it again. It turns out the botanists that study this, oh, there it is. The botanists that study this point out that the origin of a thorn on a rose bush is very much associated with the epidermis of the rose bush, and a botanist who's very let's say a guy's got a PhD in botany, he would not call this a thorn in the same way as Doki Doki asked about the thorn on a cactus. It turns out they come from two different parts of the plant in the sense of the outermost part of the, let me silence my own phone here. The outermost part of the plant in the stem. You remember from high school, college botany about the xylem and the phloem and all the different parts of a woody plant stem. The true thorn comes from a different area than what we call a thorn on the rose bush. Thank you, Doki Doki. And by the way, I'm sorry, I didn't really answer. I have worked in the desert east of San Diego, and so I've actually been out around these cacti, and I've seen where animals have gotten in and eaten at the base of them. I think the answer to that is that in our modern world, at least, those thorns don't protect the cacti from being eaten. They certainly are an annoyance to the descendants of Shem, Ham, and Japheth as they came over and had to exist in a desert environment. Those cacti thorns got them on a number of occasions. Okay, next.
Very good, David. Doki's got another question. Do animals really evolve horns and antlers to fend off violent predators?
Oh, excellent. Excellent question. So now we're going from botany to zoology, and we're asking the question, well, why do deer and elk and moose, I guess, why do they have antlers? Are they trying to make sure that the hunter does not run toward them with a spear and they can gouge them with the antlers and so forth? No, I think it has nothing to do with that. I think that God designed creatures like elk and deer to have the potential to have horns. And because I live in a culture here where my own relatives and a bunch of my coffee drinking buddies pride themselves in killing a deer. In our modern world, it's called harvesting a deer. I don't really, I think we'll just say that they kill the thing. When they go out to get these deer, they often find that the antlers are beaten up, not from predators trying to get the deer, but these are mating ritual fights between a couple of bucks uh, in the in the woods. Um, I'm part of a Bible study that meets every Wednesday at 5:30 in the morning, and one of the men that's in the Bible study is a rancher. He has all kinds of cows, and there's a certain ratio of how many bulls you should have with how many female cows. And he made an interesting comment to me. He said, I always keep 3 bulls. And my logic of this is that during mating season, 2 of the bulls will be fighting each other as to which cow he can marry. And that frees up the 3rd bull to go ahead and mate. So I thought that was an interesting way the modern rancher deals with this. Now, they're not there to fend off predators. They're there to fight off who they're going to mate with.
Very good, David. I appreciate the responses to these great questions. Okay, next question. Can flood geology explain the formation of caves better than secular geology? As you know, Professor McQueen, one of the primary objections to young Earth creation is that caves, and specifically stalactites and stalagmites, take millions of years to form.
Yeah, that's a very interesting question. I'm going to use an analogy using the Lincoln Memorial in Washington, D.C., and also the Jefferson Memorial in Washington, D.C. When I worked for the United States Geological Survey, we lived for 4 years in the D.C. area, so I visited these monuments and seen the answer to this. But let's start with the idea of caves and have a word about stalactites and stalagmites. In my misspent youth in Tennessee, one of my hobbies was spelunking, cave exploring, which I went with my buddies. So I've seen these with my own eyes. The 2 words stalagmite and stalagmite, one of them has a G in it. And that's how if you look at a cave and you see a formation coming up from the bottom, that's a stalagmite coming up from the ground, and stalactite coming down from the top. I have seen these in limestone caves in Tennessee, and I often wondered how long it took them to form. Well, if we change our picture here and use a different color like this green color, so this is not a cave now that someone can stand up in like this. Let's imagine this to be the basement of, let's say, the Lincoln Memorial or the Jefferson Memorial in Washington, D.C. Well, the floor and the support of both of these places are made of marble. And we certainly know when the Lincoln Memorial and the Jefferson Memorial were erected. Well, if you go down to the substructure to actually look at how the engineers designed the foundation of these 2 wonderful monuments in Washington, D.C., you'll find that if you go down in here, you have the same sort of thing that you see in a cave. In other words, in both of these memorials, there are stalactites and stalagmites that have formed in decades, not millions of years, in these caves. One of the lesser known books to a lot of people that I'm having some problems with my computer here. I've got to take a break here and get some power to it. But is the audio and visual, is the audio and the video still coming in good, Donnie?
Yes, it looks great. If you need to get a power source, we can move to our break after you're done answering this one.
Okay, let me answer this question, then I'll go put some power to this machine. So we know when both of these monuments were built, and so we know that it did not take millions of years, but rather decades and at most 100 years for these stalactites and stalagmites to form in the basement of these United States buildings in Washington, D.C. And so what I will do is add some humor to our discussion. Since here in the U.S. we just passed the 4th of July holiday, it's time to say, down with George III. We're ready to have American independence. So if you'll go ahead and drop my video, I will go find some power and be back in just a moment. Okay, Donnie?
Okay, David, I appreciate the answer. We will take, feel free to take your 5 to 10 minute break. And as David is on break, I will go over some reminders and announcements. So firstly, I still have several questions to get to, which we'll get to in the second half of this program. And we got a lot of events coming up. And so I've been booking events left and right. I've got events booked into September, as a matter of fact. So July is basically fully booked. We got a ton of debates, ton of shows. We've got quite a few open mics as well. And so I am incorporating more and more open mics on the various topics that we cover, because what this allows us to do is present on a specific topic, but then open it up to whoever is interested to basically join and engage the topic. And that keeps it interactive, and that keeps it from being an echo chamber, right? We can get people in here who disagree with us. They can ask questions, present arguments. And so that's what we'll be doing for the next 2 nights. I am pumped for tomorrow. Myself, Sam, and Kent, we will be together for this evolution open mic night. We will start the show off like we usually do. First 20 minutes or so, we are going to address some arguments. Specifically this time, I'll be pulling up a BioLogos article on the so-called best evidence for evolution. We will engage those arguments. Specifically, we'll have Kent refute those and respond to those. Our last open mic, we've been doing these evolution open mics about once a month. Time flies by. So I feel like we just had our last one, and we debunked Professor Dave and his evidence for why he's related to strawberries, banana plants, and whales. And so if you haven't checked out that one, that was a ton of fun. We went for about 4.5 hours, lots of great impromptu debate, lots of great impromptu discussion. Okay, so then the very next day, we are going to be having another open mic, this time on a totally different topic. It'll be on the Transcendental Trinity. And we'll have Matt Yester here. Sam's also going to be here as my co-host and moderator. And so Matt will be giving a presentation on this topic. And what's interesting about this topic is it will apply to those that reject the Trinity, the biblical Trinity. And so skeptics of Trinitarian theology are welcome to join and challenge Matt. This will be a fun open mic. But also atheists, those that reject God in general, because Matt will be presenting on why the Transcendental Trinity best explains reality, and why the Trinitarian God is essential. And so skeptics of all flavors are welcome. Next month, we've got Paul Price, who will be joining me for an open mic. This one is going to be This one's going to be awesome. I'm excited for this one. So he'll be presenting on historical science versus operational science. And immediately following the presentation, we're going to open up the mic for anybody who disagrees or anybody who just has a question for Paul. Feel free to join, and we're going to have some fun. So I'm just looking at the questions in the chat. Andrew Cumming, appreciate your question. Andrew Cumming joined our last open mic, and we had some great discussion on pseudogenes, synonymous mutations, nested hierarchies, ENCODE, junk DNA, so on and so forth. And so Andrew asks, just curious, why did you decide to post one particular segment of our discussion in genetics? Okay, so if you go to the video section, I uploaded a great clip of a portion of our 4.5-hour open mic where Matt and I engaged Andrew specifically on synonymous mutations and pseudogenes and the nested hierarchical patterns that they form and so on and so forth. And so if you haven't yet seen that, please do check it out. I do have the open mic in its entirety linked in the description box for people to check out. So the reason why I posted that is because those arguments presented by Andrew are some of the go-to arguments for a lot of evolutionists these days, especially a lot of the PhD evolutionists or more informed critics. And so I think it's important that people see us as creationists engaging those talking points. And I think that is a video that demonstrates how young Earth creationists can respond. So appreciate the question. And we did, in that open mic, we had other portions where we talked about pseudogenes, for example, when Kent was there. And that is another great portion that'd probably make a good clip too. But to see all of it in its entirety, check the open mic that goes about 4.5 hours. So a couple more shout outs. We have a lot of eschatology and dispensationalism related debates coming up. So these topics have been some of our most viewed topics. Our audience really enjoys these. I enjoy them. And so I've done a lot of work reaching out to people who enjoy debating this topic in order to have a lot of good engagement on it. And so we got Matthew 25 parables, definitely a controversial topic, lots of different opinions on this. And so Charles Jennings, Pastor Tommy McMurtry, they'll both be here next month, or I should say this month, now that we're in July. June flew by. The 10 virgins, the talents, the sheep, and goats. That's going to be a great discussion. We've also got a couple full preterism debates. Actually, I just booked a big one between William Bell and Dr. Sam Frost. And so I am pumped for this one. Not this one specifically on the screen, although I am pumped for this one, but the one that I'm talking about, William Bell and Dr. Sam Frost. I just confirmed the details, and so I still got to get a thumbnail for it and start promoting it. That's going to take place in August. But the proposition for that debate is going to be the resurrection of the dead occurred in 70 AD. That'll be William Bell as a full preterist. He'll be taking the affirmative, and Dr. Sam Frost, he'll be taking the negative in that debate. We got another full preterism debate here, Stacy Turbeville and Pastor Scott Clem. And so this is coming up this month. Pastor Scott Clem's got a couple debates, as a matter of fact. He'll be debating not just Stacy, but he'll be having another debate with Merritt on Daniel's 70th week. And Scott's also going to be joining us for an eschatology panel discussion where I've got several people, Sam Frost, Scott Clem, Phelan, who has a great channel discussing all things eschatology, and Pastor Anthony Aquino. So this is just going to be an eschatology a night of discussing all things eschatology. David McQueen, I'm not sure if you can hear me. I see you backstage, but your screen is all black. And so I'm not sure if you got your power source plugged in time, or if your webcam accidentally went off. Feel free to let me know. Okay, so I think his power, or maybe he's just restarting it. So once I see David join us again, I'll bring him in. Okay, another couple discussions we have coming up. Matt Slick and Caleb Percher. They'll be debating, more of an informal debate. I'll be guiding in as moderator, the Trinity versus monarchianism. Matt has debated here many times, 12 or 13 times. Caleb's also debated here several times too. And so I think this is going to be a fantastic discussion. Coming up next week, Dan Schappa, Matt Slick, discussion on unlimited atonement. We're also going to be engaging the critics. We're going to be engaging the chat as well. And so anybody who wants to join or send in questions, please make sure you're here for this discussion. Another big one coming up in August on dispensationalism. David Preston, Pastor Matt First, is God done with Israel? So controversial topic, always a fun topic to debate. We at Standing for Truth Ministries, we've been putting on I haven't seen too many other channels putting on these kinds of discussions on dispensationalism and Israel and eschatology. And so I'm really looking forward to all of the ones coming up over the next couple months. Important topic, lots of different views in the world of Christianity, whether it's pre-mill, post-mill, amill, whether it's preterism, partial and full. Within the pre-mill camp, you got your pre-trib, your post-trib, your pre-wrath. And so we're coming together and we are discussing these issues the best that we can. And so David just let me know that his computer is booting up, and so he'll be back in 5 minutes. And so we'll see him in about 5 minutes. We've also got Charles Jennings and Cabane, or Seraphim Hamilton. They are going to be debating the topic of justification by faith alone. And so we've also got some big soteriology related debates coming up for you, including this one. And I'm currently working on confirming details on a couple others. For those that love the Trinity debate, we do got a formal debate here. Carlos Xavier and Kelly Powers. Does Matthew 28:19 teach the Trinity is biblical? And so that's just a snapshot of the overall material that we do have coming up for you in the next couple months. If you're new to this channel and this community, but you love debates on all sorts of topics, check the debate playlist section on the channel. I've now hosted over 330 debates, I believe, on all sorts of topics. So whatever your favorite topic is, I'm sure there's at least a dozen debates on that topic for you to enjoy. Doki doki. Thank you, brother. Check out the website, endless content there as well. On the main page to the right, there's a whole massive section of just most common questions, arguments, criticisms, objections. And I've got links in there for people to find quick and easy answers. You can also check out our podcast. Currently, I've got 130 plus episodes uploaded, and I am looking for help. If anybody knows how to work a podcast and has done it in the past, I'm looking for help to get more of these podcast audios uploaded. Okay, so Professor McQueen, good to have you back, brother.
I apologize. I lost power on my computer, but I've corrected that now. And so let me summarize the paleobotany argument from Scripture, and then we'll go on to other questions. Okay, Donny?
That sounds great, David. Take your time.
Okay. If you go back to the third day of creation and read it from the standpoint of paleobotany, this is what you come up with. On the third day of creation, you have the formation of grass. Then the next category is herb yielding seed. And then the third category of botany is fruit trees yielding fruit after his kind. Now, let me actually read that in Genesis. And we can see what the truth of Scripture says. This is reading from Genesis 1, and we'll go down past the second day, and we'll go down to the third day of creation in verse 9. And God said, let the waters under the heaven be gathered together into one place, and let dry land appear. This is the argument for one world continent in this creation week. And God called the dry land earth, and the gathering together of the waters he called seas. And God saw that it was good. And God said, let the earth bring forth grass, the herb yielding seed, and fruit tree yielding fruit after his kind, whose seed is in itself upon the earth. And it was so. Now notice that this resolves several questions that the evolutionary community often throw up to us. Oh, McQueen, yours is the chicken and the egg argument. Which came first, the chicken or the egg? Well, this answered it from a botanical standpoint. Listen to what it said. Yielding fruit after his kind, whose seed is in itself upon the earth. And so it was so. So the first peach tree already had a seed in it. And so this is the chicken and the egg argument turned around dealing with botany. And then in verse 12 it says, and the earth brought forth grass, herb yielding seed after his kind, and the tree yielding fruit, whose seed was in itself after its kind. And God saw that was good, and that was the evening and the morning of the third day. And so we can actually turn to modern botany and also paleobotany to see that the plants that we find in the fossil record do actually fall into these three categories. We have some evidence of grasses, herbs yielding seeds, fruit trees yielding fruit. In the future, when Donny brings his family down here to Louisiana, we will be able to go about 2 hours away from where I live to find a fossil bearing unit that has leaves in it, very much like these leaves, that are pressed down and are easily identifiable as to their genus and species. And so we can see that these plants have not evolved. They were knocked down and buried during the time of the Great Flood. I went and got some props, Donny, to better answer some of the questions earlier. May I turn to that now?
Definitely. Yep.
Okay. Here is Gentry's book called Creation's Tiny Mystery. And on the front of this, you see a radioactive halo. Now, notice the number of rings that you see there in the alternating light and dark areas. If I turn the book around and bring it up close, these are polonium halos. And you'll notice that there are fewer rings here than here. It is that diametral section, and I think you can see a point of light there in the middle. That would be a fluorite or a biotite. Gentry, I think, anticipated in the 70s that people would argue that the radioactive halos were not primordial, but secondary from some other decay process. And so in 1973, Gentry wrote a paper in Science. I'm sorry, this one was from Nature, a refereed journal. And here is the title and first few sentences. Ion microprobe confirmation of lead isotope ratios and search for isomer precursors in polonium radiohalos. Radiohalos associated with the decay of several polonium alpha emitters have been studied by optical microscope, mass spec, an ion microprobe. In such cases, a large excess of lead 206 compared to lead 207 was found to be incompatible with the radiogenic decay from uranium 238 to uranium 235. This has to be explained on the independent formation of polonium radiohalos separate and apart from the uranium decay chain. Now, many of you who are students of American history would recognize a mica sheet like this. I have some thinner sheets down in here. But if you have studied, whether it's Canadian history or American history, about these types of mica, the old German word for it is isinglass. And if I pull off a piece, which has fluttered off on my computer there, you'll see that this is transparent. And this was used for window glass in Pioneer America. Let me get this piece off my system here so it doesn't cause me to go offline. The black equivalent of that is biotite, and that's where Gentry got his specimens. Okay, we can go back now to other questions that you might have.
So, if I could clarify this ongoing debate, even within the young Earth creationist community, Brother McQueen,
Yes.
on the origin of these halos. Both, if you could speak to this a little bit, both groups within the young Earth creationist community would agree that radio halos formed rapidly, but one group would say that this occurred during the flood from accelerated nuclear decay and uranium decay. Is that right?
Well, excuse me. My appreciation of Andrew Snelling's counter arguments revolving around radioactive halos, and maybe I need to go back and reread some of the things he's written about it, but my appreciation of what he says is this: especially the evolutionary critics of Gentry's work will build their argument in this way. They will say, here you've got a pegmatitic structure, and let's say that this distance is 1 meter. And in this pegmatite, you have got the mineral biotite, the mineral fluorite that's in the matrix of this pegmatite. Their argument is that over here, a few meters away from this pegmatite, is a uranium to lead decay chain. In the uranium to lead decay chain, polonium is part of that product, that decay process. So their argument is that fluids move through cracks in this pegmatite and in the fluorite and the biotite, and that the polonium 210 and 214, 218 is introduced in that way. Well, there are two enormous problems to this. One of these isotopes of polonium has a high half-life of less than 3 minutes. In no evolutionary scenario can you imagine the movement of these fluids in a matter of minutes over into this pegmatite. The second problem with this, which I've actually seen with my own eyes in looking at the examples that Gentry had me work with 50 years ago, you should be able, and let me get a different color here. you should be able, in a fluorite, let's take this one and blow it up and look at it here. You should be able to see if there are cracks and veins through which the polonium could be introduced into this crystal structure. And many of you know that I've written a book about mineralogy called the Mineral Book, and I've received training over 50 years in crystallography and mineralogy, and you just don't see this. There's no supporting evidence. It is what you, Donny, and also George have taught me over the last 2 years to call a rescue device. This is a rescue device that the evolutionary community uses to explain away the polonium halo argument.
And so they really have no good reasoning from their evolutionary starting point for this evidence directly in the rocks for accelerated decay, whether it's at the flood, creation, or both.
Yes, and let's think through the logic here of what these individuals are saying. They are saying that in the evolutionary model, a muscovite crystal like this that solidifies inside of a granite, for example, would take a very long time to cool. And what they overlook is a type of geochemical process that is called eutectic crystallization. And that is, if you have the temperature and the pH exactly right in a mixture of silicates, you can get the rapid formation of a mineral. It's called eutectic crystallization. And so even using their own petrology, their own geochemistry, they ignore the fact that something like this could have been formed very quickly. Now, Gentry does build on that by saying, and he argued this in the early 80s with Dalrymple at the Arkansas Creation Evolution trial. He argued with me, he said, you know, you cannot produce a granite the size of my fist in a laboratory because they're miraculous. Well, Dalrymple's argument was, oh, Gentry, what you've introduced us to is just a tiny mystery. And that's where this book got its title, from Dalrymple's comment to him. I don't fully understand it, Dalrymple said, but it's a tiny mystery. It's a minor thing, because we know that we can take pulverized quartz and pulverized feldspar and pulverized mica and put them in a very small capsule, less than a cubic centimeter, and we can put it under high heat and pressure, and we can produce a glass which has the chemistry of a granite, the bulk chemistry. And Gentry's argument, you know, he's passed away now, but his argument still holds. Well, that's all well and good, but produce for me a granite the size of my fist. And Dalrymple dismissed that as being beyond the scope of technology.
Very good, David. A very interesting topic, and so I really appreciate your insight and your thorough answers. Is very helpful. Okay, so I guess we'll move to the next question. One of your biggest fans, Professor David McQueen, is here in the chat to support your presentation. Gracious crybaby. Question for David. If not from natural selection, why do we find blackberry bushes in very dense thickets where they are protected from herbivores?
Okay. I have to pause on that because I have a couple of blackberry bushes in my yard here in Louisiana, and they do tend to be in an area of growth. But what I find is an herbivore like myself can reach my hand in and still get that blackberry out. And then what makes me mad is that the local birds, the creatures that are here in Louisiana that love berries, they come in and they eat my blackberries before I can get to them. And so these dense thickets that you're proposing here, crybaby, are certainly found in the woods here in Louisiana, and they have thickets where there are indeed thorny plants. Let me switch briefly to the biology of black bears and brown bears, grizzly bears. One observation that wildlife biologists had made for years was that these bears would go into, on purpose, a kind of a thicket where there were thorns and briars and all that kind of stuff. And they would lunge into them, actually jump into them, and kind of roll around, purposely getting pricked by these thorns and thistles. And the wildlife biologists wondered, well, what in the world is going on? Well, it turns out that in many of these thickets where the bear, which is an herbivore, could be eating the blackberries and so forth, they would jump into these areas in order to roll around, and the little pricks would give them immunity to certain disease. And so it's not a function of natural selection in an evolutionary sense, but rather a process that the ICR scientist, Dr. Galuza, has come to call engineered, and his exact phrase slips me right here at the moment, but it's kind of like
environmental tracking.
Yes, thank you. And engineered environmental effects that were there in those first two bears from 6,000 years ago and have been passed down genetically to today.
Okay, David, thank you for the response. Another one from Creationist Crybaby. And he says, David, isn't it more accurate to call rose thorns prickles? Thorns are derived from shoots. Prickles lack the vascular tissue of shoots and are extensions of the epidermis. Any thoughts on that, David?
Oh, well, I thank you so much, Cryberry Baby, because I should have looked that up. Only to a geologist would this on a rose be called a thorn. The correct word is prickles, because the prickles lack any vascular tissue in the shoots, and they're extensions of the epidermis. So thank you for clarifying that. I had read about that, but I didn't make it part of my initial preparation. Thank you, sir.
Thank you, David. Next question, common objection, comes up in almost every flood debate. How can a catastrophic worldwide flood, like the Genesis flood, preserve things such as raindrops, dinosaur egg nests, and footprints? So basically, they'll argue that these very delicate preservations should have been washed away like dinosaur egg nests. Or I think they'll even say, how during a flood would dinosaurs have time to lay eggs and have these egg nests and so on and so forth? What's a good response, David?
Okay. In the time we have remaining, which is about 30 minutes, I think I'll just pick one of these, and that's the footprint argument, and build on that.
Sure.
Because this is something that I've seen with my own eyes. There are a number of places from Texas all the way up through central Canada, where you can find dinosaur footprints. The locality that I know best is about a 5-hour drive away from me to the west over in Texas, in an area south of Fort Worth that is called the Paluxy River. And so this Paluxy River has been of interest to National Geographic and a number of people for coming up on 100 years, because in earlier issues of National Geographic, they had photographs in there of and it's called the Glen Rose limestone in the Paluxy River, south of Fort Worth. And in the Glen Rose limestone, you can see trackways. And one of my favorite photographs is during a year when the Paluxy had a very low volume of water anyway, some creationists went down and actually sandbagged it enough and swept off a portion of the riverbed. And in it, you can see three-toed dinosaur footprints going this way, and then human footprints. And I'm talking about with an obvious toe and arch and heel going this way. And so here you have the human footprints, and then here you have the dinosaur footprints. Now, some of my creation colleagues have said, McQueen, you should not even talk about that because it's hard for us as creationists to explain, as it is the evolutionary community. Well, the late John Morris, I think, had the best idea about this. And early in his research on the Glen Rose, and in the 1980s, he actually took me with him and showed me this with my own eyes. His theory ties in the work of Dr. Steve Austin and the floating mats. And so here's kind of a cross-sectional view of what the idea is about how you could get human footprints in a worldwide flood. So the idea is that I want to draw a cross-section. Actually, no, I don't. I'm going to draw a map with this being north. And then this is a part of Texas that's called the Llano Uplift. I visited this. It's an uplifting of granitic rock, either a Creation Week rock that had been pushed up during the flood. But anyway, it's a very obvious tectonic feature in South Texas. His idea is that, combining Steve Austin's work, huge floating mats, a floating mat of vegetation began to move as the floodwaters were receding from east to west. This floating mat is something that is the size of like the state of Kentucky or the state of Delaware. This is a huge volume of matted down vegetation, all tied in together in the latter part of the flood. Remember that the teaching of Scripture, the assurance we have, is that by the time that Noah and the 7 others got out of the ark, everyone else was dead. exactly when in the flood that everyone died, that's something that we can speculate about. So the theory is that later on in the flood, as the floodwaters were receding, there were some dinosaurs on this area, and then miles away, there were some humans. This moved into this part of Texas as the floodwaters were receding. There was a change in the geochemistry of the limestone, this Glen Rose limestone, that made it like the micrites you've heard me speak about in other contexts. And so, as this big floating mat beaches itself here, the humans get off, the dinosaurs get off. One is chasing the other. They're all trying to avoid the rising floodwaters, and we get the human and the dinosaur footprints together. Now, the critic will say, "Oh, McQueen, come on. You're telling me that worldwide catastrophic currents were moving during this time and we're able to preserve this?" And the answer is yes. You can have in an area like this, and going all the way up, this is Texas here. Let's go all the way up into central Canada. You can have an area, and you do have evidences of dinosaur footprints going all the way up there. The fact that it's very rare to find the dinosaur within a few hundred meters of the footprint is a further evidence for catastrophism, because the waters would have washed the dinosaur away, leaving only the footprints. I hope that helps.
Is that evidence of these creatures during the flood basically running from the floodwaters, getting their footprints preserved, and then later on their bodies are buried?
Exactly. And this is a very unique case in the sense that in this part of Texas, with this floating mat idea, it provides an explanation for how you could get dinosaur and human footprints near each other, since it's a case that I don't think anyone in the 100 years before the flood hit would have wanted to live in an environment, in an ecology where the dinosaurs were. In the same way that I live in Louisiana, but I choose to live in a part of the state where I don't have crocodiles and alligators, I guess would be the best word, walking through my backyard.
Right. Great point. I'm curious, when it comes to these preserved dinosaur egg nests that we constantly hear the evolutionists talked about, I'm curious as to your thoughts, because from my understanding, during the flood, the crust of the earth would basically be flexing up and down. And so we would have parts of the earth that are uncovered for a few weeks or even a few months. And so during those periods of time, these dinosaurs could be laying eggs, and then eventually the flood levels rise and buries them all. I don't see why it would be an issue, David.
Yeah, and I'll respond to that. Let's take your scenario and put it on the right side of our brain. Then on the left side of the brain, we can think of something else. When the floodwaters hit, there were areas of that one world continent where dinosaurs were breeding, and there were egg nests and dinosaur eggs around and so forth. When the catastrophe struck and swept the female dinosaurs away, it's still the case where you could get the actual nest itself, the actual eggs, caught in a sedimentary unit that could be transported a great distance, then deposited in the rock record of the flood, and you would have dinosaur eggs, but no dinosaurs around.
Very good. The next question comes from me. And so the question, David, do you believe that a good line of evidence for catastrophic plate tectonics is the cold slabs, those massive slabs of cool rock that they've found at the base of the mantle, that could be evidence for rapid plate subduction during the flood.
Yeah.
If you could speak to that, what are your thoughts on that?
Yeah, and I'm so glad you brought that up, because in the next couple of weeks, I'm going to be submitting an abstract to the fall meeting of the Geological Society of America, which generally occurs in late October around Halloween, 1st of November, that period of time. George and I intend to write a paper which talks about these zones of cold rocks, also the area that is BLOB blobs coming up from the intersection of the mantle and the crust. And so I think in catastrophic plate tectonics, we have, with this evidence or theory, you might want to call it, of if we have areas where heat is rapidly rising from the standpoint of physics and thermodynamics, we have another area where the heat is being taken away. It's a heat sink. And so in previous debates, my opponents have said, oh, McQueen, you got a heat problem. You know, if you got that much catastrophic plate tectonics happening and rapid radioactive decay, you would boil the ocean dry. And they always like to go back to the Garden of Eden, and it would boil Adam and Eve and so forth. I don't think that's necessary, because I think that George and I have discovered a explanation from engineering and physics that would allow us to get rid of some of that heat.
Well, and the discovery of these cold slabs that go quite deep into the Earth's crust, as you've talked about many times, if uniformitarianism or actualism were true, and these slabs of rock got to where they are millions of years ago through snail pace movements of the continents, basically these plates, shouldn't the slabs have melted? Shouldn't they be a lot hotter if that was the case?
Yeah, it's a very good point. The geologic term that would be used is the term equilibrium or equilibrate. And so the idea is, okay, we have evidence of these cold slabs underneath, let's say, the Western United States. How come they're still cold? It would seem in millions of years that there would have been an equilibration, that the temperature would have been modified in such a way that would be the ambient temperature of the base of the crust or the upper part of the mantle, what some people call the asthenosphere. But they're not. They're clearly colder objects, and a wonderful evidence for a young Earth, because if these cold slabs were sitting around like that, they would have been brought to the same temperature as the surrounding lava.
That's a good point. Are they determining the temperature of these slabs of rock, David, because of density? They're looking at basically the density of the rocks and then also the surface rocks, and it's the same, implying it's the same temperature. Is that how they go about it?
Well, George and I, in our research, have actually struggled on that point, because a lot of the evolutionary scientists that talk about these structures, they never say anything about how they know how hot they are, or how they're able to determine the temperature of that object several kilometers down in the Earth's crust. And so George and I have chosen to make some assumptions. Like, for example, here's one of them. And our opponent in Italy, Luca, that you've arranged debates with me, with Luca over the years. One of the arguments that he has brought up is this very thing of what are the initial conditions if we're going to talk about the physics of this. Well, what George and I have chosen to do is make some certain assumptions, like pre-flood, the highest mountain was 10,000 feet. Pre-flood, you have a situation where the rocks that are being subducted and rapidly subducted and so forth would have a temperature. And in our assumption, I know that the hottest lava on Earth comes from the Hawaii chain, which is supposed to have one of these blobs underneath it feeding their magma chamber. And that lava in Hawaii is around 2,000 degrees. Now, when you get a rock that is that hot, it doesn't matter whether it's 2,000 degrees C or 2,000 degrees Fahrenheit. If you use that as part of the model, that's what we're doing. And we're assuming that these objects at depth are around 2,000 degrees and trying to work through the thermodynamics using that number.
Those are some great points. I've heard Dr. Tim Clary basically point out that they've discovered some of these slabs, David, going down hundreds of miles into the deep hot mantle. So I think the fact that they're still there and they haven't melted is evidence that they were taken down there within the last several thousand years, which is consistent with the flood model.
Clary's point is well taken.
Brother McQueen, you've done a fantastic job. We just hit the hour and 45-minute mark, and we got through all the questions. You did a great job answering them. It's been fun.
I have been doing a lot of talking, visiting my grandchildren and so forth, and I am about to lose my voice, but let me end my time with it's a funny anecdote. It's not a joke. We visited our daughter in Mississippi, who has 4 children, 3 daughters and a son who's a baby. Well, the soon-to-be 4-year-old granddaughter of mine, named Hannah, is a very adventuresome little girl and also a very willful little girl. And so here's an anecdote that I just can't help but laugh about. So my daughter pointed at little Hannah and said, "Now you stop doing that." And so Hannah, the little 4-year-old, pointed at her mama. And her mama said, "No, don't you point at me." And this is what she did. She took her other hand up and she folded back the pointy finger and brought it back to herself. I can't wait to see what this child is like as a teenager.
Amen. Great stuff, David. Was there anything in terms of concluding comments on the topic of paleobotany that you wanted to make, or did you?
I do need to summarize that. If you look at the literature of paleobotany, in my research over the last 30 days, if you go back to before about 1995, a lot of references don't even refer to the evolution of forests from predominantly fern all the way up to deciduous trees. They don't even talk about it. But those that do talk about it begin talking about it in the 21st century. But they use what would be called a rescue device. They say, even though we don't have evidence of ferns evolving into pine trees, we know it must have happened. And so they make the assumption that there were once forests in the Mississippian and Pennsylvanian period. For those of you that are international, the Carboniferous period, that produced these huge ferns that were laid down as coal deposits. And that those plants evolved with time into plants like a poplar or an evergreen. There simply is no paleobotany evidence of that. You find each of these types of plants appearing in the rock record abruptly and fully formed. The abruptness speaks of a catastrophic deposition, and the fully formed part speaks of what we read in Genesis chapter 1 about their creation on the third day of creation. Thank you for this night and for the opportunity to do that summary, Donny.
My pleasure, David. 2 hours always flies by with you, and so I really appreciate your time, David, that you give us for these shows.
And as George always says, I'm moving the cursor down, and I will bid good night to
God bless, David. You get some rest. You earned it, brother. Great show. Okay, that concludes our show on paleobotany. Did herbivore dinosaurs attack rose bushes? Lots of fun, lots of great engagement from the chat. I appreciate all the questions, the comments, and the feedback that have come in. That's why we do these live. We want to be interactive with our audience. If you have questions, whether you're a skeptic or non-skeptic, we want you to always feel comfortable in knowing that you can send in your whether it's a criticism or a question and so on. So Honesty Angel, appreciate that, sister. Hit that like button, share around this content, and stay tuned because we got a lot for everybody to look forward to. Tomorrow we're going to be back here 8 o'clock sharp, 7 o'clock central. I have already confirmed with both Kent and Sam. So Evolution Open Mic Night. We've been doing these once a month, very frequently this year. As last year, we did the 2022 Evolution Debate Challenge series. That was prolific. It was a ton of fun. We did over 60 debates in 2022 in our Evolution Debate Challenge series. So that was pretty wild. It was lively. It's a great year, busy year. We had a lot of trilogy debates during that year as well. And so if you're new to this channel, but you love a good old-fashioned debate on evolution, check the debate playlist section on the channel, and you've got endless content to enjoy. And so for 2023, we moved into doing a lot more open mics, which allows multiple people to join and provide what they believe is the best evidence for evolution. What that allows for is people who might not have been necessarily comfortable doing a formal debate. It allows them to engage in impromptu debate. And I think these open mic nights have been a lot of fun. I've enjoyed participating in them. I've had my award-winning co-host Sam join me for these. As a matter of fact, I'm keeping Sam busy. He's going to be here tomorrow, and he's also going to be here on Friday for the Transcendental Trinity. And I think both of these open mics are going to be a lot of fun. And so we've already got people lined up to join the show for these open mics. Next month, we've got another open mic. This one will be with Paul Price. Paul Price has done a lot of debates with us, discussions. He just completed a couple discussions on evidence for the existence of God. And that was with Michael, the Canadian atheist, and also Grayson. And so I think those were two really good shows. And if you haven't yet seen them, please do check it out. So Paul is happy to engage in an open mic on an important topic as well. And so I hope people will be able to join us. So before I wrap it up, I wanted to address an argument that I've seen utilized a lot. And it has to do with so-called beneficial mutations. And so we did a show about last month. I think it was one of my favorite, if not my favorite shows on mutations in general. and a thorough response to the best so-called examples of beneficial mutations out there. And I did it with Dr. Jerry Bergman. As a matter of fact, Jerry is going to be back here with us next month for a show on evolutionary phylogenetic trees. And so I think that's going to be great. Some great content there. And so he did a show with us. It was about an hour presentation on mutations. And he went through HIV resistance, antibiotic resistance. He went through sickle cell anemia, all of your most frequently used examples of so-called beneficial mutations. And then we had a roughly hour discussion where Jerry and I went back and forth on the topic. And so one specific example that I see constantly being promoted as an example of a truly beneficial mutation, the type of beneficial mutation that is required to take your fish to fishermen, basically for forward evolution. And that is nylon digestion by bacteria. And so I got a couple slides here that I want to go over. But what I've discovered is that there are many bacterial species that can actually break down plastics or these byproducts, basically nylon specifically in this case. And these bacteria, they've already got the enzymes, these pre-existing enzymes that are actually called beta lactamases, and they can break down the same chemical bond. And so basically these pre-existing enzymes could already slightly break down the bond, but due to a few mutations, okay, in these enzymes, they have now become just a little better at breaking the bond, meaning that the bacteria can now break down these plastics. This is not an example of forward evolution, and that is why I want to go over just a couple of these slides. You can find this documented in the secular literature as well. And so here's just a couple slides from the show I did with Jerry. And nylon digestion by bacteria. And so here's a couple slides I just want to go over for everybody, as I do get this question coming in. And so I wanted to address it here. Nylon digestion by bacteria. So this is not the story of a highly improbable frame shift producing a new functional enzyme. This is the story of a pre-existing enzyme with a low level of promiscuous nyelinase activity, which improved its activity toward nylon by first one, then another selectable mutation. So basically they're saying here that the origin of nyelinase actually provides a nice example of the optimization of a pre-existing fold's function, not the innovation or creation of a novel fold. A protein that already has a low level of activity for a particular substrate can be mutated. So this is a pre-existing enzyme. It can be mutated to favor that side reaction over its original one, often in just a few steps. So one could even argue that it's a loss of substrate specificity. And right here, bacteria from a diverse set of environments have the pre-existing ability to digest nylon waste products. So bacteria in differing environments than the bacteria that are found in this environment that allows them to digest nylon, which is man-made, only came about recently. They also have these enzymes. They also have the pre-existing ability to digest some of these waste products. And so it's really not a surprise that a couple mutations can allow them to digest these byproducts slightly better than they did previously. And so this is not the type of mutation that is going to explain large-scale evolution. But most importantly, this is not the kind of mutation that is going to counterbalance the damage done through the influx of all of these low impact, these nearly neutral and very slightly deleterious mutations that are pouring into the genomes of living organisms. And so I like to point out that even if you had 10, we can sit here all day arguing about whether or not sickle cell anemia or HIV resistance or the human lactose mutation, if these do truly provide a novel feature or genes to these organisms. You can have 10, you can have 20, you can have 100, but you're going to need many more than that to counterbalance the damage from all of these mutations that are pouring in. And the human mutation rate is roughly 100 new mutations per person per generation. And so a few beneficial mutations is simply not going to counterbalance that damage. The Lenski experiment demonstrated that beneficial mutations, they're rare. They're about 1 in a million, but typically when they happen, they're reductive. And so one thing that I've noticed with the evolutionary community is they almost always want to document the effects of these mutations based on the phenotype, right? Sickle cell anemia, for example, has an effect that allows those with sickle cell anemia to be resistant to malaria. Well, sickle cell anemia is bad. You don't want sickle cell anemia. That's degenerative. And you also don't want malaria. But having these deformed hemoglobin molecules or proteins basically allows for resistance to malaria. But when you actually look at the genotype, what's going on under the hood? So rather than the phenotype, the physical features of the organism, let's look at what's going on a genetic level. And you'll find that it's typically reductive. With sickle cell anemia, it's due to a broken gene, a broken protein, a broken cell, essentially. And so this is reductive. We've seen this in Lenski's experiment, where the several adaptations that have been observed, they are actually due to these bacterial species. They are losing genes. They're losing genes short term for adaptive purposes, but it's actually long-term degeneration. It's very similar to if I was driving my car and I wanted to get better gas mileage in that moment. Well, I can start removing weight from the car, right? I can start throwing things out. I can take the mirrors, throw them out, remove the seats, remove the steering wheel. Yeah, so removing weight from the car is going to temporarily give me better gas mileage, but it is not doing anything in terms of being an overall benefit to the car, because it comes down to net gain versus net loss. And the evolutionists, they'll oftentimes point to a trade-off. They'll say, oh, there's a trade-off because these mutations are environmentally dependent. It's context dependent. And so sometimes you get a mutation that's deleterious, but then eventually you'll get a beneficial mutation that's just that's so good that it's basically going to counterbalance that damage. But if you're having all of these deleterious mutations pouring into the genome, okay, and they're just tons of these nucleotide sites are being degraded, they're being destroyed, and you're just waiting for just a single desirable beneficial point mutation, let's say. Well, by the time you actually get that so-called beneficial mutation, you've already had thousands of nucleotide sites being degraded. And so all you ever get with your so-called trade-off is shrinking functional genome sizes, basically. And that's what we see in Lenski's experiment, are shrinking functional genome sizes. And so I'm working on just a full comprehensive presentation on all things mutations, where I want to address all of your best so-called examples. And once I'm ready with that, with my slides, I will present that. And so I want people to understand, though, that it is important that not only are we looking at the phenotypic effects of these mutations, okay, we need to actually see what's going on a genomic level. What actually led to this change? Because the examples that we see of beneficial mutations, you're not going to climb up a mountain by continuously taking steps downward. Okay, you actually need to take many steps upward. And so that's what we see is most of these mutations that are claimed to be beneficial, they are overall, they are reductive. Okay, they're functionally compromising to the organisms that experience them. And so I've heard it explained by a young earth creationist geneticist. It's called The House and the Happy Wife. And so basically, Dr. Kevin Anderson, he explained that because your wife wants a larger dining room, you as the husband decide to knock out an interior wall to make the dining room bigger, right? But you don't tell us how the house was built or originated. because all you're doing is knocking out pre-existing systems, and that's not actually telling us how those systems got there. And so nobody would tell a carpenter that the house was built by knocking out internal walls to make for that larger dining room. And that's what we see on the genotypic level with these kinds of mutations, is we see downhill changes, where the creation model would say that God basically placed us up at the very top of the mountain, and deleterious mutations over time have moved us slowly off the mountain. But evolutionists want us to believe that we can climb Mount Improbable. We can climb Mount Improbable through mutations that are overall very deleterious to systems. So in a presentation I'm working on, I'm going to focus on the HIV resistance example that Dr. Bergman touched on, nylon digestion, of course, human lactose tolerance. Dr. Bergman and I discussed that one. That's another big one that's oftentimes brought up, is the human lactose tolerance example, and then also the nylon digestion, which obviously doesn't work, as we've covered here. But with human lactose tolerance, we actually have a lactase gene that's shut down during puberty, which means lactose in milk cannot be digested. And so most people are lactose intolerant. They do not make that lactose gene after post-puberty. But some people actually have a mutation in what's called the MCM6 gene, which regulates the lactase gene. And since the gene is hit with a mutation that controls the lactase gene, that lactase gene is never shut off. It's like a light switch that's just permanently on now. Okay, it's never shut off. And this mutation actually keeps the MCM6 from shutting down the lactase gene, meaning people with this mutation can now digest lactose. And so if we are to do what I said, look to the genotype, not just the phenotype. Genotype wise, this is a mutation that destroyed a pre-existing system. And so systems are being lost. Systems are breaking down. These mutations are overall, they are functionally compromising to these organisms. And so you're never going to climb Mount Improbable by continuously taking steps downward. You have to take steps upward. I see. Okay, so I am going to wrap this up here. That was kind of impromptu, but I did want to address it as it's oftentimes brought up in our open mics. The nylon digestion. Matt Powell, brother, good to see you. He says, this is such an excellent presentation. I appreciate the kind words, and you keep up the great work. Love it, brother. Okay, pseudonym. Super chat. $5. Thank you for the support. Would you consider a similar species of different color a beneficial mutation? So I guess, if I'm understanding correctly, you can take bears. I think there's 8 species of bears. You've got polar bears, black bears, brown bears. Your polar bear, in that specific environment, the Arctic environment, it has a white coat color. And from my understanding, that's basically due to just a point mutation that has allowed it in pre-existing genes that has allowed it to adapt to that environment and adapt rather rapidly. Okay. And this actually comes down to the concept of epigenetics, this layer that is above and beyond the genetic code, basically. Okay. And we've got sections of our DNA. The sections of our DNA can basically be tagged with a methyl group, right? The carbon atoms, which allows the cell to turn a gene off or turn a gene on, depending on the environmental conditions. And so we got millions of genetic switches in the genomes of living organisms just waiting to be, again, turned on or off, depending on your environment. And so depending on your environment, you could grow taller, you could grow shorter, or in the case of what we're talking about here with coat color, you can have mutations that are oftentimes deterministic, because we know mutations are more non-random than random. And so God knows. He has divine foreknowledge. He knows the end from the beginning. He knows the environmental conditions that are going to pop up in the post-flood world. And so he has front-loaded organisms with the pre-existing ability to adapt, to change, And so I think a lot of these mutations are actually determined in the sense that God knows what environments creatures are going to go into after the flood. And so some mutations might actually be favored over other mutations. And in the situation of coat color, you can have a bear, right? You've got a bear archetype on the ark, and from those pre-existing DNA differences created heterozygosity, but also that pre-existing ability to adapt to various environments. You have a bear species that goes into a colder environment, the Arctic environment, and whether it's through a non-random mutation or whether it's through epigenetics or recombination, where you can get those pre-existing DNA variants to shuffle up every generation, leading to variation and diverse, even novel phenotypes can come about that way. And so these are built in, basically, and God has allowed organisms to adapt. I like to say ever-changing environments require ever-changing genomes. And so we live in a world with ever-changing environments, and therefore the ability to change, the pre-existing ability to change, is important, or else species would go extinct after a few generations if they were not able to adapt. Okay, let's see here what else we got. Alec Cox mentions the HIV. So basically HIV resistance. That is a big one that's used as well. And when you first hear it, you might think, okay, phenotypically, that sounds like a truly beneficial mutation. No reductive effects at all. But, and Jerry and I discussed this, he presented on it, and he made some fantastic point. Basically, there's a small number of people that are resistant in this world to HIV, and they have a mutation in the gene that makes what's called the CCR5, and they no longer have it on their T cell. Okay. And so this means that the HIV virus cannot bind to that T cell. It essentially doesn't have the key to get in. It can't bind. And that's what Dr. Bergman was talking about. These viruses require certain keys to infect or bind to these cells. And so this HIV virus cannot bind that specific T cell. And genotypically, a pre-existing protein has been completely knocked out. And therefore, we have this phenotypic effect where a small number of people are resistant to HIV. But again, this comes down to the fact that it's reductive, it's functionally compromising overall, and this mutation doesn't tell us where the gene came from. It doesn't tell us where the pre-existing genes that are hit with these mutations, or the pre-existing regulatory systems that are being hit with these mutations, or that are being rearranged. It says nothing about where they came from. And that brings me back to the illustration I was talking about with the house and the happy wife. Okay. So your wife wants a larger dining room, and me as the husband, I decide to knock out an interior wall to make that dining room bigger. Well, you're never going to say that you got the house, you built the house, the structures within the house, the bricks, everything came about by knocking things out, by knocking down pre-existing systems, by destroying that interior wall. No, it doesn't tell us anything about the origin of the house or the building blocks for the house. And so the point is, even your best examples of beneficial mutations, when you actually examine them and study them comprehensively, they turn out to be reductive. That's why, again, don't just look at the phenotypic effects. Also look at the genotype. What's going on a genetic level? And you will quickly realize that it's typically due to the breaking down of pre-existing systems. I'm not saying that's necessarily always the case. There might be a rare example that when you examine it on the genotype, there's no reductive effects. But again, that rare example is not going to do anything to counterbalance the damage that we are witnessing in terms of beneficial mutations. Pseudonym says, that makes no sense. You're arguing for beneficial. Well, again, it is quote unquote beneficial because you do have some kind of adaptation being observed, like with sickle cell anemia. Yeah, the people with sickle cell anemia have a resistance to malaria. But again, malaria is bad, sickle cell anemia is bad. Overall, it's degenerative. Overall, it's reductive. It's functionally compromising. It's due to broken systems, broken proteins, broken cells. Okay, and so if that's what you mean by beneficial mutation, it's like saying if you had a row of people and the police were handcuffing everybody, and so you decided to cut off your hands. So now you can't be handcuffed and you can run away and escape. Okay, well, I guess it's beneficial in that moment, in that context. But the second you run away and you leave that situation, you're now left without hands. Okay, so overall, it's degenerative. It's breaking things down. It's not good. Or you decide to cut off your feet in order to not get athlete's foot. Okay, in the context of getting athlete's foot, great. Now you don't got to worry about getting athlete's foot, but you're going to have to worry about all sorts of other problems now that you've cut off your feet. You no longer have feet, and so that's what we see in the so-called beneficial mutations. Is yes, we see in their various contexts, we see a benefit to be had, but overall, it's due to a loss of some kind. It's due to degeneration. It's kind of hard to quantify information. I typically avoid saying, well, these mutations are the result of a loss of information, because it can get a little bit tricky defining information or quantifying, what was lost specifically, how much information was there previously, how much information is now there because of the mutation. It's best to just point out that these mutations, when they occur and you examine them on the genotypic level, okay, you actually look at the pre-existing genes that are being hit, look at the pre-existing systems, you see that they're typically being lost, they're being wiped out, and so it's reductive. It's functionally compromising. You almost don't even have to go down the information route, because it's not necessarily a matter of loss or gain of information. It's about the evolutionists having to provide us with a mechanism that can explain the origin of these sophisticated, these very elegant systems that are found in the cell. And so if these mutations are mostly reductive, they're mostly damaging and degenerative, they're never going to be able to provide us with the necessary mechanism that will help climb Mount Improbable, which is what the evolutionary community wants us to believe happened. And so they are actually forcing their interpretation of what they believe, the evolutionary story, onto the fossils. When they look to the fossil record and you see these historical reconstructions done, you oftentimes see this with evolutionists who'd rather focus on phylogeny and the fossil record rather than genetics. They are forcing their interpretation or what they believe about the evolutionary story onto the data, because if we don't actually have a mechanism today that we can observe that can drive evolutionary change, then all we get with the historical reconstructions is a story, because it doesn't fit reality, since they can't provide us with a mechanism that's going to explain large-scale evolution. There has to be genetic events that demonstrate how you can construct highly complex genetic systems, and we don't see that. We really don't. Pseudonym's question here, let me see, I think is another super chat. Is it better to have sickle cell in pain or die? Okay, well, again, we would have to look at, oh, my phone's ringing, just 1 second. We'd have to look at what's going on, again, on the genotypic level, as I've said. So malaria resistance, sickle cell anemia. Okay, so having sickle cell anemia guards people from getting malaria, and malaria can kill you. Okay, so sure, you might rather have sickle cell anemia, this disease, over getting malaria and dying. But again, when we actually examine what's going on in the genetic content of those that have sickle cell anemia, it is due to a broken gene. It's due to a broken protein. It's due to the breaking down of pre-existing systems. It's a deformed hemoglobin molecule, basically. And this is reductive. And so malaria kills you. Sickle cell anemia leaves you with this disease. It leaves you functionally compromised. And so, again, it's not necessarily about gain or loss of information. It's about being able to explain how we are going to be able to counterbalance the damage done by the majority of mutations that are coming in. They're being passed on generation after generation. They're nearly neutral. Selection can't see them. They're slightly deleterious. And over time, species decline genetically. They descend into genetic sickness. And that's never going to take your pond scum into people. That's never going to take your single-celled like ancestor into a whale over time. It's never going to take your fish to amphibian to reptile to mammal to man. Degeneration is never going to explain the origin of these highly complex and sophisticated systems. So I think I've gotten to all the questions. And so I hope people enjoyed this. Honesty Angel, appreciate it. Yes, much needed haircut. Now I'm good for the summer. It's getting a little long and hard to manage. So Jessica B says, right. I appreciate it. Okay. So anyways, we're going to wrap it up. We're coming on 2.5 hours. And that's a sneak peek into some of the work I'm doing on beneficial mutations. I think it's an important topic. And I think if we can show everybody, whether evolutionists or creationists, that their best examples of beneficial mutations, when we examine the genetic content of what's going on, it's reductive. And even if you have a rare beneficial mutation that's not reductive in any way, it's so rare, it's probably going to be lost over time. It's not going to be fixed. It's not going to do anything to counterbalance the damage. Then creation wins. It's a great time to be a biblical creationist, everybody. So Doki Doki, great to see you, brother. Lots to look forward to. Lots of debates, discussions, interviews, conferences, lectures. We're doing it all here on Standing for Truth Ministries. And so with that, God bless all. Actually, go check a video I just put out this morning. Not a lot of people, I'm not sure if a lot of people know it's there, but I put it out this morning. It's on mutually interdependent systems. So I'm going to do a series, going to go through a bunch of different examples on how these chicken and egg problems, there's 2-way and 3-way chicken and egg problems, how they preclude naturalistic origins. And so in this video, I focus specifically on DNA repair systems and the fact that we have the genome is hit with just so many mutations every single day. As a matter of fact, we have roughly 1 million new DNA lesions in every single one of our cells every single day. And so with 50 to 60 trillion cells in our body, that's a lot of damage we are experiencing daily. And so I talk about how we have these DNA repair systems that are built into the genetic systems of living organisms that actually fix these DNA breaks. Okay, and there's a chicken and egg problem associated with these systems, DNA and DNA repair enzymes. And I put out a video, it's only about 12 minutes. The answers I've gotten from the evolutionists is hilarious. You know, basically all you have to do to respond to their answers is cool story, bro. So evolutionists, they hope, dream, and imagine. They love their stories. And with that, God bless all. Standing for Truth is out.